Welcome viewers, welcome to the new lesson from Dr. Arts Biology. If you did not subscribe the channel, you can just click on the subscribe button and bell icon. Welcome students. So far we were discussing about unit 1. Now we are just moving into unit 2 that are nothing but discussing about macromolecules. In that unit 2, the first topic is the macromolecule carbohydrates. So in this series we will be just discussing about the carbohydrates and different parts or different topics under the carbohydrates. Hope you have learned the unit first in a well manner and it's thorough with you. It's better to just complete the first unit and then move into the second unit. Okay, later otherwise you will be having confusion. As it is biochemistry, you will be just dealing with so many structures and metabolic pathways. And if you are just accumulating the content to study later just before the examination, what will happen is you will be having a lot of confusion and you will not be in a state to clear the examination well or you score the mark well. So in order to avoid all those things, better study one by one in a systematic way. Now we are just moving into the topic carbohydrates. Okay, so first of all, we have to learn about the basic structure of carbohydrates, and before that, let us see what is actually meant by the carbohydrate and all. So we can see here, carbohydrates are, we can say, most abundant organic molecule. So the most abundant organic molecule on earth is carbohydrate. We can see the two cases. We know that is cellulose and then chitin, which form the first and second most abundant organic molecule on earth. Okay, so this is the first and second most abundant carbohydrate, and we can say cellulose, we know that it is a part of the cell wall of the plants, and chitin is actually the part of the exoskeleton of lower organism. So especially in case of the insects or arthropoda, for example. Okay, so these two are the carbohydrates which are most abundant. You can say the organic material, organic molecule that are most abundant on earth is these two. Okay, now the carbohydrate is actually playing both metabolic and structural role. So carbohydrates in an organism, carbohydrates plays both the metabolic role as well as the structural role. What is meant by metabolic role? We know that different types of metabolism is happening in our body. Then metabolism is nothing but the overall chemical reactions that is happening inside your body. That is what we mean by the metabolism, and that we have classified it as the anabolism and the catabolism. Anabolism and catabolism, where anabolism is a constructive process where the molecules are synthesized, and catabolism is the degradative process where the molecules are break down in order to release the energy, and that energy will be utilized for continuing the life. So carbohydrate plays a major role as a metabolic part. It is involved in the body's metabolism. In addition to that, we know that it is also playing a role as a structural component. The two most important example is nothing but the cellulose and chitin. So they become the structural part of the body. So you can see that cellulose is a cell wall. That means it becomes the structural part of the cell. Chitin is become the structural part of the exoskeleton. We found our hyaluronic acid, which is normally found in case of the cartilage and all. That also is actually functioning as or playing a role in the structural component of different organisms. So there is a basic introduction regarding the carbohydrates. The most abundant organic molecule on earth, cellulose and chitin are example for that. But cellulose plays the first role and chitin is in second role. It plays both metabolic and structural role in different organisms. Now we have to remember one more thing. Animals they can synthesize the carbohydrate from, they have synthesizing carbohydrate from lipid. They can synthesize it from glycerol. They can synthesize it from amino acid, etc. So if the carbohydrate is not derived directly from the food, they will be actually utilizing the stored form in their body, in animal's body, just like a lipid, glycerol, amino acid, etc. And they will be just converting these compounds, these biochemical compounds, into carbohydrate. So animals are able to do that particular role or particular biochemical reactions which actually form the carbohydrate from lipid, glycerol, amino acid, etc. And we will be just calling the process as the gluconeogenesis, and we will be just discussing that in metabolism. But ultimately, all the carbohydrates that are coming into the animal is actually from plants. We know that plants are the only things which are capable of producing carbohydrate, synthesizing carbohydrate, and animals ultimately depends on the plants for the carbohydrate. Okay, so that's about the basic introduction regarding the carbohydrate. Now we can just move to the basic structure. What are carbohydrate? Which are the chemical components of carbohydrate? How it is actually different from each other chemically? All those things we will be just discussing now. Now let us move to the basic structure. So let us see the basic structure of the carbohydrates. We can see that carbohydrates are actually polyhydroxy carbonyl compounds. So carbohydrates are actually polyhydroxy carbonyl compound. What is mean by that particular thing? What is actually polyhydroxy? You can see that hydroxy is nothing but those which stands for the OH group. That contains polyhydroxy means this particular compound contain many number of or many hydroxyl group in it. That is what actually mean by the carboxyl. So the polyhydroxy carbonyl compounds. Let us see what is mean by the carbonyl. Carbonyl is nothing but the aldehyde or ketose. So these are the two functional groups of carbohydrates. So the aldehyde is nothing but the one which stands as CHO, and the other one is C double bond O. So this CHO will be exactly looking like C double bond OH. So this is actually the carbonyl part. So they are, carbohydrates are polyhydroxy carbonyl compound which contain either aldehyde or ketose and multiple number of hydroxyl group. Okay, so that's about polyhydroxy carbonyl compound. Now we can see that most of the carbohydrate will be composed of carbon, hydrogen and oxygen, and they will be present in the ratio 1:2:1. Okay, so all the carbohydrates contain carbon, hydrogen and oxygen, and most of them will be satisfying the ratio 1:2:1. So we can say that the general formula is CH2ON. That means carbon, hydrogen and oxygen will be formed in 1:2:1 ratio multiple times. That is what actually mean by the carbohydrate. But at the same time, you have to keep in mind that this particular formula will be satisfied only by the monomers, and it cannot be satisfied by the disaccharide, polysaccharide, etc. or any derivatives of carbohydrates will not be satisfying this particular general equation. So we cannot say that all those compounds which are not satisfying CH2ON does not belong to carbohydrate, or we cannot say all carbohydrates satisfy the general equation CH2O. Yeah, there are deviation from this particular general equation, but we can say that most of the carbohydrate will be satisfying this particular general equation. Okay, so this carbohydrate can have derivatives, just like we can say more mucopolysaccharide. Mucopolysaccharide is actually a derivative of carbohydrate which will not be satisfying this particular general equation. For example, for mucopolysaccharide is nothing but the chondroitin, heparin, etc. And at the same time, we can see if an amino group, if an amine derivative is the carbohydrate, you can see that it is like a glucosamine, galactosamine, etc. This will be containing extra hydrogen, so it will not be satisfying this particular general equation. So we have to keep in mind there are molecules which belongs to carbohydrate, but that doesn't actually follow this particular general equation CH2ON. Okay, so let us now move to the more details of carbohydrate. So we can see that carbohydrates are actually classified based on the functional group into two. Okay, so classification of carbohydrate we are just moving, that also belongs to the basic structure. So based on the functional group, we can just classify carbohydrates into two, that are aldose sugar and the keto sugar. So based on the functional group that is present, carbohydrate can be classified into aldose and the ketose, or otherwise known as the aldoses and the ketoses. What is aldose sugar? The carbohydrate which contains the functional group, aldehyde functional group, the CHO. And the keto sugar is nothing but the carbohydrate which contain the keto functional group, the C double bond O, the keto group. So based on this functional group, we can just classify it as the aldose sugar and the keto sugar. Okay, now let us see how can we classify the carbohydrate based on the complexity. How many number of units are present, how complex that particular carbohydrate is. So based on the complexity, we can just classify the carbohydrates into monosaccharides, then oligosaccharides, and the third one is the polysaccharide. So based on the complexity, how many number of sugar units are present, then the carbohydrates are classified into monosaccharide, oligosaccharide and polysaccharide. So we can see here, monosaccharide will be composed of single sugar or one sugar unit, and the oligosaccharide will be having 2 to 9 sugar unit, and the polysaccharide will be having 10 and above. So 10 and above sugar unit will be there in case of the polysaccharide. We will be studying these in detail one by one. So I am not writing an example and all those things here, but we will be just discussing monosaccharide separately, oligosaccharide separately and polysaccharide separately. So the complexity, based on the complexity, it is divided into monosaccharide, which is composed of only single sugar, oligosaccharide 2 to 9 sugar units, and polysaccharide, which will be composed of more than 10 or more than that sugar units. Now let us move to the monosaccharides. So what is actually monosaccharide and all? What are the main things that we have to learn under monosaccharides? So you have to keep in mind that I am just following your syllabus only. Kannur University, BSc Zoology, 5th semester, Biochemistry and Endocrinology syllabus only. So the content delivered will be just as per the syllabus. If you want to learn more about this particular thing, you can just watch my other videos that is uploaded in different playlist. So you have to keep that in mind. I'm just delivering the content exclusively meant for your syllabus, not more than that or not less than that. Okay, so keep in mind that particular thing. So monosaccharides, we know that monosaccharides are the carbohydrate which contain only one sugar unit. So here we can see that monosaccharide we can just further classify based on the complexity. So based on the complexity we can just classify monosaccharides into different groups. It is actually based on the number of carbon. How many number of carbon is present in monosaccharide? Based on that fact, we can just classify monosaccharide into different groups. Normally, the number of carbon is actually 3 to 7 in case of the monosaccharide. If there are 3 carbon, it will be known as the trio sugar. If there are 4 carbon, it will be known as the tetra sugar. If there are 5 carbon, it will be known as the pentose sugar. And if there are 6 carbon, it will be known as hexose sugar. And if there are 7 carbon, it will be known as, you can just tell it, you know this particular tri, tetra, penta number. So it will be known as the heptose sugar. Okay. So we already learned about the aldose and the ketose sugar. So in each monosaccharide there are aldose derivative and the keto derivative. There are aldehyde sugar and the ketose sugar. Okay, under each category of this particular monosaccharide. For example, if I am just classifying that particular thing, so I am just writing aldose here and ketose here. So I am just writing the example that you have to learn as per the syllabus. There are a lot of examples. If you want to learn all the structures of this particular monosaccharide, you can just watch the video that is already uploaded in my channel. You can just see in the description box, or you can just see here as in the form of card. Okay. So this particular trio sugar, the aldose is nothing but glyceraldehyde. Glyceraldehyde sugar. And the ketose is nothing but dihydroxyacetone. The both aldo and keto under trios you have to learn. We will be just discussing that later. Now, the next one, the tetra sugar. Tetra sugar, there is erythrose. This is easy to remember the keto sugar of erythrose. You can just make it as ulose. Then it will be keto sugar. So, erythrose, keto sugar, your keto derivative is nothing but erythrulose. Okay. Now, the pentose sugar. You are familiar with the pentose sugar. What is an example for pentose sugar? You are learning it from grade 12 onwards, because it is there in the DNA. We know that DNA or RNA is actually ribo or deoxyribonucleic acids. So that ribo component, that is actually a pentose sugar. That is nothing but, you are familiar with that, and that is nothing but the ribose. So ribose is an example for pentose sugar. And there is an easy way to remember the ketose, because you just add ulose there. Then it will be keto sugar. So ribulose is actually the keto sugar. So you don't have to learn erythrulose and ribulose, because it is not mentioned in your syllabus. I am just giving examples only. Now let us see hexose sugar. So we have a common hexose sugar. You are familiar with that because we have learned glycolysis and all in grade 11. So it is nothing but the glucose. Okay, and what is the keto derivative of glucose? Is nothing but the fructose. you have learned it as the sweetest sugar on earth, that is present actually on in fruits, in case of the honey, etc. So the sweetest sugar is the fructose. So in honey you can see the levorotatory form of the fructose, that is L-fructose, and that is nothing but the levulose. We'll be discussing that later. So glucose and fructose are examples for the hexose sugar. And now the heptose is there. So heptose is only one example that you have. So pseudoheptose, and the keto derivative is nothing but the ulose. So it is pseudoheptulose are the keto derivatives. So this also you don't have to learn, and so ultimately you have to learn the structure and functions of monosaccharides such as glyceraldehyde and dihydroxyacetone under triose sugar. Now under tetrose, erythrose, and then under pentose, ribose, and under hexose both glucose and fructose, and under heptose you don't have to study anything. But you have to just keep these particular examples in mind. You don't have to study that in detail. Okay, so these are the main monosaccharides. Okay, based on complexity, we have classified it as a triose, tetrose, pentose, hexose, and heptose, where the number of carbon is actually 3, 4, 5, 6, and 7 respectively. Now let us see some aspects of this particular monosaccharides. So let us see that. So monosaccharides, there are different isomers of monosaccharides. Okay, there are different isomers of monosaccharide. We know that isomers are nothing but the compounds whose chemical formula is same, but the configuration is different. The spatial organization of atoms or molecules within that particular is different. That is actually what mean by the isomers. There are different isomers that is seen in case of the monosaccharide. You don't have to learn in detail about those isomers. You just want to touch that and know what it is actually. Okay, so only that much is there in your syllabus. So I am just touching what are the different isomers in case of the monosaccharide, and we are not going into details of that. As I told you before, if you want to go into details, you can just see the other videos that is uploaded already. Okay, so let us see the monosaccharides. There are mainly in case of the monosaccharide, the most important one is actually the D and L isomers. So D and L isomers. When you see D and L isomers, you may be having connection with the dextro and the levorotatory, but it is not at all related to dextro and levorotatory. That particular thing is actually D and L. It is not a capital D and L. So this is entirely different. So let us see that in order to understand what is actually D and L isomers, let us see one example. Let us see the erythrose structure. So erythrose, we can see this actually the aldehyde sugar. So CHO will be there, and we can see that there are 4 carbon, 1, 2, 3, 4, and the 4 carbon will be having hydroxyl group. So OH, H, OH, and CH2OH will be there. So this is actually erythrose. Now what kind of erythrose it is? D or L erythrose? D or L erythrose? This particular thing is actually the D erythrose. Okay, so D erythrose is this particular thing. So keep in mind what is actually mean by the D erythrose. You have to touch this particular thing, OH. Okay, if this particular OH is there on the, what is this particular side? In case of the molecule, if there is on the left side. In case of the molecule, if that OH is on the left side, then it will be not this particular OH, this one, penultimate OH. So if that particular OH is on the left side of the molecule or right side of you, let us see the left side of molecule, because we have to tell or we have to describe it based on the molecule. So if this OH, in what way we can define this OH? We have to just consider the functional group first. From the functional group, ultimate carbon is what? This one is the ultimate carbon. So carbon before ultimate carbon, what it is actually? Penultimate carbon. So this particular carbon is actually penultimate carbon. Okay, if that OH group is on the left side of the molecule, then it will be known as the D-erythrose. Okay, and if that OH is on the right side of the molecule, okay, right side of the molecule, not with regard to U, then it will be known as the L-erythrose. Okay, so you can just see the L-erythrose, the CHO, CHOH, COH, H, CH, OH, H. So if the penultimate carbon hydroxyl group is on the left side of the molecule, it will be known as the D-erythrose, and if it is on the right side of the molecule, it will be known as the L-erythrose. That is what actually mean by the D and the L isomers, and it is actually defined based on the hydroxyl group that is attached to the penultimate carbon. So don't forget the name of this thing. This is penultimate carbon because it is just before the ultimate carbon from the functional group. So that's about D and the L isomers. Now let us see about the another isomer, that is nothing but the epimers. So let us see that. So epimers. Epimers are nothing but the monosaccharides which differ in their configuration only at one carbon except penultimate carbon. Because we know that penultimate carbon is we already utilized it for the D and L isomer. So are monosaccharides which differ in their configuration except one carbon. Okay, so only at one carbon except the penultimate carbon. Okay, that is what actually mean by the epimers. In order to understand the epimers better, we have to learn the monosaccharide structures, and you can just see the video. That is, I have given the link there. So you can just see the video and then you will understand how to identify epimers and all if you are preparing for the competitive examinations in order to get into the PG program and all. Okay, so you can just watch that particular thing. I am just sticking the epimers here in a brief way. So they are the carbons, or they are the monosaccharides which differ in only one carbon configuration. So let us see. So I am just writing, for example, the ribose and the arabinose. Okay, so ribose and arabinose I am just writing. So ribose structure, we know that the CHO, CHOH, 5 carbon is there, 4, 5, OH, H. Then I am just writing the D ribose. So HOH, then HOH. Okay, so this is actually the ribose sugar. And if I am just writing the arabinose, okay, so ribose arabinose, you can just see that it also has the same thing, CHO, C, 5 carbon, 2, 3, 4, 5. Okay, and it will be just differing from this particular ribose only at one carbon. So you can see here, so penultimate carbon will be having HOH, because I am also writing here D ribose and D arabinose. Okay, D ribose and D arabinose I am just writing. So this will be differing only at one carbon, and that is at one only, the second carbon. the first carbon from the functional group. So you can see here the hydrogen will be present here and OH will be present on the right side of the molecule. Okay, so you can see here the entire structure is same for ribose and arabinose except at the carbon 2. So in carbon 2, in case this OH is on the left side of the molecule, but here OH is on the right side of the molecule. So except this particular carbon, the arabinose and ribose are same. So we can just say that ribose and arabinose are epimers. Now what epimer? You have to just mention the carbon number. So we know that this is the first carbon and this is the second carbon. So at which carbon the configuration is different? The second carbon. So we can just say that ribose and arabinose are C2 epimers. Okay, so that's about epimers. Keep in mind, epimers are monosaccharides which differ in their configuration only at one carbon except a penultimate carbon. So penultimate carbon we are utilizing it for the D and L isomer. So that's about the epimers. As your syllabus is just mentioning that only, I am just touching it. If you want to go into detail, as I told you, you just watch the other video. Now let us move to the next isomer, that is anomers. In order to understand the anomer, we just want to learn about the ring structure or ring form of monosaccharide. Okay, so in order to understand that, I am just taking the glucose example and how it will be forming the ring structure. So you can see here, the glucose is having 6 carbon, 3, 4, 5 and 6. And the first one is actually functional group because it is the aldose sugar, so CHO. And it will be CHOH. In case of the glucose, the third carbon will be having OH on the right side of the molecule. H, OH, H, OH. Okay, and then H, OH, H. Okay, so now you just count the number of asymmetric carbon, chiral carbon in this particular glucose. So this is actually the D-glucose. What is meant by asymmetric carbon? The carbon whose 4 valency is filled by 4 different atoms or molecules. That is what mean by the asymmetric carbon. If there is an asymmetric carbon, 2 raised to n isomer will be there. If it is 1, 2 raised to 1, that means 2 isomers will be there. If it is 2, then it will be 2 raised to 2, that means 4 isomers will be there. Now let us count the number of asymmetric carbon in case of the D-glucose. So you can see here in this C, the 2 valency is filled by the main oxygen itself. Therefore, that first carbon is not a asymmetric carbon. So this is not asymmetric carbon. Now take the case of this particular C. So you can see that H, OH, CHO and this particular thing. So this one is actually asymmetric carbon. This one is one. Is this? No. Why? Because 2 valency is filled with the hydrogen. So the glucose is having 1, 2, 3, 4 asymmetric carbon, 2 raised to 4, 16 isomers is possible in case of the glucose. Okay, now let us go to the ring structure. The ring structure is actually formed when this particular hydroxyl group is actually attacking this particular carbon. So a bridge will be formed in between. So as a result, the structure will be, the ring structure will be looking like this. C, O, there is another C, 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 C. Okay, so 1, 2, 3, 4, 5. You can see that 1, 2, 3, 4, 5. So the fifth carbon OH is attacking. So this O is actually this particular O. And there is a double bond O that is left. And this H will be combining with that. As a result, it will be forming like this. Okay. I'm just drawing it in the opposite way. So HOH. So this first carbon will be having HOH like this, because actually it was HC double bond O, but this hydrogen came there, as a result OH is there, and this hydrogen left this place, that's why only O is there. Okay, now where is the sixth carbon? Sixth carbon will be just projecting from this fifth in the form of CH2OH. Okay, clear? And it will be having one hydrogen here. Now this one HOH, you are having third carbon here, so third carbon OH will be different, and the remaining H and OH is different. Okay, so this is the pyranose ring structure of glucose. Okay, so this is how pyranose ring structure of glucose will be looking. Now let us count the number of asymmetric carbon in the ring form of glucose. So let us see. So this was the first carbon, which was actually non-asymmetric carbon, and we can see here this is the first carbon. Now let us see whether it is symmetric or asymmetric. We can see that one bond is filled with hydrogen, one bond with the hydroxyl group, one with oxygen, and one with the CHOH, and the remaining portion. Okay, so we can see that the 4 valency of this particular carbon is now filled with 4 different atoms or molecule, and as a result the first carbon is now asymmetric carbon, which was actually a non-chiral carbon, which was not an asymmetric carbon in case of the linear structure, but that turned into an asymmetric carbon in case of the ring structure. So a new asymmetric carbon is formed there. Now the isomers resulting from this new asymmetric carbon is known as the anomers. Okay, so anomers are nothing but the isomers, okay, which are resulting from the newly formed asymmetric carbon when the linear structure turned into a ring structure. And there are two types of isomers that is resulting from this new carbon, anomeric carbon. This carbon is now known as the anomeric carbon. So let us see what are they. They are nothing but alpha and beta isomer. So alpha anomer and beta anomer. Alpha anomer and beta anomer. So what is meant by alpha anomer? If this OH group attached to the anomeric carbon is below the plane of the molecule, then it will be known as the alpha anomer. So we can just say that hydroxyl group attached to the anomeric carbon is below the plane of the molecule, then it will be known as the alpha anomer. And what is actually meant by the beta anomer? The opposite. If that asymmetric carbon hydroxyl group is above the plane of molecule, then it will be known as the, okay, so now we can see that OH is above the plane of the molecule, then it will be beta anomer. Okay, so you should be familiar with the anomers now. Anomers are nothing but the isomers that are formed from the newly formed asymmetric carbon, okay, when the linear form is changed into ring form. And this newly formed asymmetric carbon is known as the anomer. The isomers resulting from that anomeric carbon is known as the anomers. And there are two types, alpha anomer and beta anomer. Okay, alpha anomer will be having the hydroxyl group below the plane of the molecule, and beta will be having hydroxyl group above the plane of molecule. So this one is actually alpha glucose and this one is beta glucose. So that's about the anomers. Okay, so if you want to see how this particular ring structure is formed and all, you can just watch my next video. It's not related to this your content, because you don't have to learn how it is formed and all. As per your syllabus, it is not covered. So I'm not representing anything here. I'm just representing the linear structure and ring structure just to describe what is anomer. Okay. So keep in mind what is anomers. So that's about the basic idea regarding the monosaccharide. Now, as per your syllabus, you have to learn detail about the glyceraldehyde and dihydroxyacetone under the trio sugar, and erythrose under tetro sugar, ribose under pento sugar, glucose and fructose under hexo sugar. So all this will be just described in the next lesson. Okay, but I hope you understood the concept. If it is not clear, you have to revise the video again. If you are not clear, you can just comment in the comment box. Then we can have a discussion more. Okay, so that's about the lesson now. You can just watch the next lesson for the next topic. Thank you.